Well, thanks everyone, and thanks for joining us. I'm extremely excited about this one particular panel. We're here to talk radiotherapies, and who would ever thought that we would be spending as much time on radiotherapies maybe 10 years ago. But clearly there's been a huge reemergence in the space. We've seen a number of acquisitions by a number of large pharmaceutical companies, most importantly led by Novartis, subsequent to that Lilly, a few others like AstraZeneca and Bristol. And with that, we've seen now a new crop of companies come and try to solve this precision medicine question, should I say. And really we're gonna learn a lot more about the different approaches that people take here and what's next. And before we go into that, maybe Umar, how has this impacted your practice and maybe a little bit more but from the clinician side?
Sure, thanks. So I can tell you that as a nuclear medicine physician, that nuclear medicine is really undergoing a renaissance right now. And it's really been driven by approvals from the FDA over the last 5 to 10 years. There's new detector technology. And the radiopharmaceutical space is just growing phenomenally. So just as an example from an imaging perspective, the PSMA imaging agents now, we routinely see prostate metastases that are 1 or 2 millimeters in the retroperitoneum, and it's driving some of our colleagues crazy because they don't know quite how to deal with that, and I think it's really changed urological practice. And so, at MGH, what we've done is we're actively pursuing radiotherapy. So we're adding 4 new theragnostics rooms. We're adding SPECT CT scanners to increase our dosimetry so we can do precision dosing for theragnostics. And we see this as a wonderful time for incredible growth in this space. And I think everyone's feeling this in the Society of Nuclear Medicine and other physician groups.
And thank you, Umar. And I guess I'll kick things off with Matt Roden. Just quickly, Matt.
Which one?
You kind of brought up this notion of this construct of RLTs being a lot like ADCs and being truly precision oncology. Maybe you want to just touch on that, and then maybe what you're doing at Actis.
Sure. So, hi everybody. My name is Matt Roden. I'm the CEO of Actis Oncology, which is a company focused on the next wave of targeted radiopharmaceuticals. And when we founded the company a couple years ago, it was really with the view that, hey, the ADC space, antibody-drug conjugates, had created a lot of patient impact, and that there were a lot of new treatment opportunities, new options for clinicians treating the patients. And so we saw an opportunity with targeted radiopharmaceuticals to really build on that and to create maybe a different angle of patient impact. And that's because if you can deliver a radionuclide into a tumor safely, then this can create a very significant efficacy opportunity for these patients. In part because, different than an ADC, the payload of cell killing is completely different. It's non-cross-resistant, and so in previous studies by some of the pioneers in the space, you mentioned Novartis earlier, that some of the pioneers, academic and industry pioneers, have shown that the depth of efficacy or anti-cancer response that you can get with radionuclides can even exceed, sometimes significantly exceed what is observed with the ADC space. And so we saw an opportunity recognizing that the vast majority of the effort being put into the field of radiopharmaceuticals is really focused principally on 2 targets. One is PSMA and prostate cancer. The other is SSTR2, which is in a variety of neuroendocrine tumors, particularly GI tumors. And really almost all of the effort has been there. There's also some more recent effort around the target called FAP. And this is really important for patients that have disease that expresses these markers. And it's wonderful that there's new and improving treatment options for the patients that have disease with those markers. However, recognizing that cancer is a collection of dozens of different diseases, and that we've only scratched the surface with these 3 targets that where most of the focus has been. And so what Actis Oncology did was we took a step back, recognizing some of the optimization, some of the most recent and more successful approaches that were ongoing in the field of radiopharmaceuticals, particularly those that minimize systemic exposure of radionuclide, in other words, outside of the tumor. We took a step back and tried to redesign the construction of how these things are made to really maximize the ability to really get into the tumor, stick around and irradiate the tumor, but then really clear out of the body as quickly as possible so as to minimize exposure to things like the bone marrow and circulating white cells and red cells. And so we really felt that there was an opportunity to do that, but even more so, what we did is we selected a set of technologies that would enable us to really open up the target space. And so we have 7 programs in development, and none of them are against SSTR2 or PSMA or FAP. These are all against other tumor markers expressed in some big tumor types like lung cancers and breast cancers and colorectal cancers and urothelial cancers and cervical cancers. And so these are really important patient impact opportunities because what we're doing is we're taking what's been learned from a relatively narrow set of patients and expanding that to a much broader patient population to drive that benefit. And so we really think that we as a field, we're all working together in different ways. There's probably many different ways to create an effective radiopharmaceutical. There's so many patients that are in need, and we're all going after different targets with different approaches. And so it's something that I'm gratified by my colleagues here on the stage, that we're really at the foot of the mountain here, right? We're in the very early part of the game. And if you think about, just again, coming back to the corollary to the antibody drug conjugate space, I mean, hey, there's been a lot of value created in the ADC space over the last 10 years as the world figured out how to optimize them and to take up as many different targets as possible. And we're in the infancy of the field practically. I think we've just now started to figure out how to optimize these things. The technologies are getting better. Supply chain is getting better. And now we're all working together here on the panel to expand the positive patient impact potential of this class of drugs.
And I think this stage represents so many different wonderful approaches from the companies on this stage in terms of ligands and isotopes. Maybe you could talk a little bit about how your companies approached the different ligands and the different sizes and why you chose the biological constructs you did. I'll leave it to anyone.
You want to start in any particular area?
Who's going to take that?
Okay. So Matt Vincent, Chief Business Officer at Advanced Cell. So the isotope that we work on is like Perspective. AIM is sitting a couple chairs away. Is lead-212. So lead, like actinium, is an alpha emitter. It is a much shorter-lived isotope relative to actinium, but also has the ability to put a lot more dose rate in the initial dose into the tumors. I think in terms of ligand selection, I'll come to in a moment. I think that one of the pieces I just wanted to add on to what Matt Roden was just saying about ADCs versus RLTs is there is an aspect of distinction here, and that is ADCs typically kill by immunogenically silent or not very immunogenic forms of cell death, right? They interfere with tubulysin and cytoskeletal elements. They bind to DNA and cause apoptotic-like mechanisms. The advantage to the RLTs, and this is where I'll come to why lead was exciting to me and what we've picked as the ligands, is that well, most people think of betas as single-stranded DNA breaks and alphas as double-stranded DNA breaks. The reality is that alpha particles can create a significant number of reactive oxygen species in a very short path length in the tumor. And the generation of those ROS species in the tumor results in things like lipid peroxidation and results in ferroptotic or other immunogenically active forms of cell death, right? So I think that is take all of what excites you about ADCs and then now add an immune component to it in terms of what these can do. Lead has a half-life of 10.6 hours, so we have a different PK and biodistribution profile that we manage against actinium, for example, with a half-life of substantially longer of days. So in our case, we tend to reach for small molecules, including organic molecules, peptides, cyclic peptides, starting to look at a couple of single domain proteins for binders. But our ligand selection tends to be driven more by this 10.6 hour half-life, where we want something that goes into the tumor relatively quickly, can deliver that very high dose rate of alphas, and then at the same time is because of the decay profile, is ultimately cleared by the body over the course of a couple of hours to 10 hours. So that's what goes into our ligand selection. Again, in our case, it's dependent on the isotope.
Yeah. I would add to that lead-212 as an alpha emitter actually has a isotopolog pairing with lead-203, which is actually a SPECT imaging isotope. So I think perhaps the major disruption that RLT can offer, as opposed to, for example, ADCs, is that you can, in advance, calculate the therapeutic window of your drug before you've even dosed a patient with a therapeutic. So you can take the imaging analog product, it's going to distribute in exactly the same way through the kidneys, it's going to accumulate in the tumor in exactly the same way as the therapeutic product. So you actually can de-risk a whole development program on the basis of 5 or 6 patients and the imaging that you can do. So I think this is something that perhaps ADCs and other targeted therapies just can't offer. The idea that upfront you can actually deliver information that will drive your whole program and also make the FDA quite comfortable with your development program. And so, you know, calculation of the potential toxicities, where the radiation is going to deposit its dose, how much dose you can get into the tumor, et cetera, can all be calculated up front with this pairing of lead-212 and lead-203.
Yeah, and I think building upon that is a great difference between the RLT space and the ADC space, which is very quickly you can see how your drug is going to work before you're actually giving the active pharmaceutical ingredient, which is the radioisotope that can emit these different particles that we've talked about to destroy the cancer cells. So it's a major advantage, I think, in this space, and I think what's driving a lot of the interest in the space. I think, so I'm Lana Janes. I'm one of the co-founders of Dera Therapeutics, which is a radiopharmaceutical company. We were founded in Vancouver and are now based in South San Francisco. And the approach, we were founded from the approach of exactly like we were talking about, how do we build a platform where you can optimize the therapeutic index? Because at the end of the day, we like to say at our company, you can't beat physics. Those radioisotopes will deliver their payload. They don't have to be internalized like ADCs. They will be delivering their particles, which is the payload that can cause damage to the, the DNA in the cancer cells. It's all where they deliver it. And so we're all, this entire panel, we're all focused on how do we best control that therapeutic index so that we get the most time on tumor and in tumor, and the least amount of time with exposure to the healthy tissues as these radiopharmaceuticals circulate. And the approach that we've taken, we were built around the notion of how do we build an optimized platform that can go after a lot of different targets and that is modular in nature so that we can actually try and develop several therapeutics. And I think our approach has been, we have a platform called ROVR, and I like to think about it, it stands for Radio Optimized Vector Engineering, but the best way to think about it is ROVR, it's a bit like a good dog. It goes where you tell it to go, hopefully, or it's like the Mars rover where it's really charting new territories. And what we've done is, through this modular approach, we've been able to nicely show how we can tune to different radioisotopes that have different half-lives and also different targets with different target densities, different types of tumors, and have a very predictable and controllable therapeutic index. And that's really what we've been focused on. And I think what's important for this room to understand is we are at the infancy of, let's call it the second generation of radiopharmaceuticals, the first one being a few decades ago. But radiation therapy has been one of the 3 pillars of oncology treatments, right? Since since we began treating patients. And so it's just a novel way for us to be able to control the therapeutic index and deliver the radioisotope dosing in a much more controlled fashion. Our first program is going into the clinic this year in small cell lung cancer, targeting a specific antigen DLL3 that's expressed on a large number of the small cell lung cancer tumor types. So what I would say is all of us as a field are pulling for each other. When one company does well, we all do well. That rising tide raises all of our boats. So we're really, this is a very tight ecosystem. I don't think there's any one right or wrong way to do it. Some of us are working with peptides, some of us are working with small molecules to get that tumor on and tumor in approach. Some of us are working with full-size antibodies. Some of us are working with smaller format biologics. And that's good. I think diversity is good for the field because it will, you know, just like cancer is not one disease, I don't think there's going to be one answer for the different types of cancers that we're treating.
Phil, do you want to touch on yours?
First of all, I'd like to say that this is a very exciting time. You know, I'm not going to come out and say that radiopharmaceutical is good, ADC is bad, or anything like that. I think there's going to be opportunities for all these modalities, T-cell engagers, CAR T-cells, to have a place in the armamentarium of oncology care. One other advantage, though, of radiopharmaceuticals are, although I prefer the agents to be internalized rather than remaining on the surface, the fact that they don't have to be cleaved is a real advantage over ADCs. So by way of introduction, my name is Phil Kantoff. I'm the co-founder and CEO of Convergent Therapeutics. My background is as a medical oncologist. My last job before I joined Convergent Therapeutics was the chair of medicine at Memorial Sloan Kettering Cancer Center. I saw the opportunities in radiopharmaceuticals, and moreover, the clinical data that had been generated by Neil Bander and Scott Tagawa at Cornell, and was very compelled to become a part of that. So Neil and I founded the company back 3 years ago. So our approach, as you heard, is an antibody approach. It's an antibody that's been around for a while. It's called J591. We know it as COMVO1 now, coupled to actinium-225, which is another alpha-emitting radiopharmaceutical, which has a longer half-life. Matt, it has a half-life of 10 days as opposed to 10 hours or 11 hours. So there are advantages to long half-life, there are advantages to short half-life, depending upon what the carrier is. So for example, actinium is not coupled with a small molecule very well. And I would argue that small molecules are difficult with actinium. So I think there's going to be a lot of combinatorial work that's going to take place, as you mentioned, over the next few years. It's very exciting. We have generated data already in over 120 patients as monotherapy and combination therapies. We see robust responses targeting PSMA and in advanced prostate cancer patients. And we see very robust responses in combinations with checkpoint inhibitors, with small molecules as well, lutetium small molecules targeting PSMA. We see synergy between the antibody and the small molecule as well. So we're very excited. We began our own IND clinical trials right now under our, rather than an academic IND, under our own IND, and we're marching forward. But as you've heard from others, PSMA is the first target, SSTR2, second target perhaps. I think there's an enormous opportunity in solid and liquid tumors to treat with radiopharmaceuticals. Thank you.
I guess as I sit here today, like one of the most interesting things about this space is dosimetry, right? And getting that aha moment of we've got something here, something that's clean from a toxicity profile, yet hitting the target. And how has that kind of elucidated a little bit more, or taken the risk off for you to a degree in terms of finding your targets, or being able to even just also get the capital necessary, right? Because all of a sudden this has been de-risked and it feels like the time horizon is much faster for your therapies. Maybe, Matt, you raised your hand down there.
Yeah, it'll be interesting to hear what other panels say. I actually wanted to use this as an opportunity, as you asked the question, about the story of when Point was acquired by Lilly, because it plays exactly into, I think, the aha moment Lilly had. Right, so I reached out to Jake Van Naarden for JP Morgan in 2023, and if you guys know, Jake is the president of Loxo at Lilly, and he said, not going to be there, not interested in RLTs. By June, walking into bio, I reached out to another friend of mine in BD at Lilly proper. They said, I said, you really should meet with us. And they said, great, we set up the meeting. When I showed up, she had brought a number of other people because they had started to see the monotherapy efficacy data that was coming out of some of the lutetium trials. A week later, we had the entire Loxo team on, but here's the eye-opener. In August, the night they gave us the offer, we had Dave Ricks in, along with the senior management of Lilly, and it wasn't us proselytizing to him, it was Jake Van Naarden. And here, he had gone from the monotherapy as obviously part of the anchor to this, to the ability to use dosimetry to make decisions in phase 1, whether you continue a program or not, right? That it was a paradigm shift in the way that oncology drugs could be developed. That you could, with a couple of patients worth of data, make a decision as to whether you continue the program or pull the molecule out and re-engineer it. And I think for those on the stage, we all know this may be new to a number of folks in the audience. FAP, which I think was mentioned earlier, FAP is a target everyone's been interested in, but no one has had a ligand that has stuck around on that molecule long enough. But you can figure that out in a phase 1 study using dosimetry, take the molecule out, figure out then the right model systems to model the way that the dosimetry data tells you a ligand against that target would behave in a human patient, and bring your second generation in, which is now what you see. To me, that's the power of dosimetry in this, in that you don't have to wait to the end of phase 2 to make a decision. You can make a decision far earlier.
And it extends into the preclinical setting as well. You obviously have preclinical cameras that can image the biodistribution of these isotopes, and so Perspective has recently announced that we'll be advancing a fibroblast activation protein peptide, because we do find that we've engineered our approach so that through multiple rounds of screening in vivo in animals. And then the de-risking event for us was 3 patients imaged in India under compassionate use to basically de-risk the program. And we saw that we did have good, very rapid uptake into the tumor, which is what you want with lead-212, 10-hour half-life, so you want to get the radiation there as quickly as possible. And then we saw good retention out to 20 hours, which means that the majority of that alpha particle radiation is actually going to be deposited into the tumor. And FAP is an exciting target because it is basically expressed in the stroma of 80–90% of epithelial cancers. So massive TAM, as we were hearing about earlier, and the idea of the potential to combine with existing therapies is one of the other aspects of radioligand therapy that I think is not yet appreciated by the market. These radioligand therapies tend to have reasonably benign safety profiles when you can compare them to sort of ADCs. So the idea of combining with a checkpoint inhibitor and these other kind of modalities is actually very attractive. Actually, we're just about to commence our melanoma program is about to combine with PD-1 nivolumab. We've recently signed a supply agreement with BMS, and we will actually be looking at dosing patients shortly with a combination of targeted alpha therapy to MC1R and PD-1. So in preclinical setting, we saw extremely exciting synergistic results, which is not always seen with these with the checkpoint inhibitors. I think a lot of people think that they see synergistic effects, but we're super excited to get that into patients who are obviously in need.
And I'm going to jump in also on the power of the imaging and dosimetry analyses. There's no question that the value of the data sets that you can generate, both preclinically and in early clinical setting, really are decisional. So give you an example, maybe connect the dots between a couple of these points. So Lana was talking earlier about how important it is to optimize the therapeutic index. We're using, at Actis Oncology, we're using mini proteins as the binders, as a targeting vector. And the reason, it's very intentional. The reason that we're using them is because they have three-dimensional shape, and so they give you the ability to hit a lot of different targets, kind of like an antibody would, but they're 1/40th of the size of an antibody, and so that means that they're very tumor penetrant and that they rapidly are cleared from the system. So these were like kind of the Goldilocks bits that we're looking for. One of the things that we found in the process of optimizing these things is, number one, as Phil mentioned, these are actually very avidly internalized into the cell, which we agree is helpful. Maybe not required or necessary, but we think it's helpful because, among other things, it traps the nuclides and the daughter nuclides in the cell. We can see, we can learn a lot about the tumor just biology, because what we can see is that once the radioligand gets into the cells, internalized, we can also see that the cell senesces and doesn't die for several days, but then finally there's a catastrophic event and the cells die and the tumor shrinks and these kind of things. So this is all things that you can learn through the imaging. Then if you jump to the dosimetry, so our lead program is targeting Nectin-4, which is a clinically validated target. It's also the target of a very successful ADC called PADCEV. And we're first in class among radioligands against Nectin-4. And we do have imaging data in patients with this agent that we're going to present at the upcoming Triple meeting in Barcelona. And I can't really say much about what those data show, but what I can say is you can learn a lot, and it is decisional as to whether or not you expect to have a therapeutic index in patients. And so we're very excited about what we're going to share there, and it really just, again, underscores the value and the power of the imaging and dosimetry approach, because even in our preclinical optimization, we took time. We said we're going to walk before we run, and we really spent extra time in the optimization of the molecule in the preclinical setting. And you can iterate very quickly by imaging the animals and seeing how much you get in the kidney, how much you get in the tumor, how much you get elsewhere. And since ours is a renally cleared agent, I think others like to avoid the kidney. We like the kidney because you can get it out of the system so quickly, but it really forced us to be really good at kidney physiology and tubular uptake mechanisms and things like that, and we figured out ways to dial that out. And so we did that all based on iteration and the power and the feedback we're getting from that imaging in animals. And it's a very powerful approach, and that's what led, again, we also, Actis Oncology has a $1.25 billion deal with Lilly as well. That's a collaboration. And it's these data sets, these optimization, these design iteration capability set that led Lilly to us in wanting to do that discovery deal to discover novel radioligand therapies against, again, new targets. And so it is a very powerful approach.
I've already told Matt, I had to sit through Jake Van Naarden. It was like an episode of the Brady Bunch. Marsha, Marsha, Marsha. It was Actis, Actis, Actis.
We have a good relationship with Lilly. Yeah.
Well, you're about to share your thoughts, I thought, maybe, on the dosimetry? No. My bad. I thought you were about to say something.
No, I would just say that, I totally agree with what everyone has said, and in many ways we're always looking for translational success to drive things forward. And in some ways, we have probably the best biomarker because we're not looking for surrogate biomarkers. We're actually seeing in a situation where we're not doing harm to the patient by administering an imaging-only version, whether it's a copy of the drug that we're going to administer later if the patient is expressing that target and taking it up. Sometimes it's identical twins and sometimes it's fraternal twins, depending upon how you switch out in the theranostic pairing. But we're very fortunate in that way that we can very rapidly not only screen for patients and see who will actually benefit from the therapy, but really be able to select those patients and then watch, be able to actually monitor them as they go. So we're in a really fortunate situation because of this theranostic pairing and improving translation all the way up.
Do you all sequence imaging before you start the therapy escalation, or how have you approached the imaging? It seems like you've all taken advantage of the companion diagnostic effort, but do you always sequence the imaging first to see to get a handle on dosimetry or distribution before you move to the treatment arm?
Well, maybe I'll jump in first on that one and then leave it to the rest of the panel. I would say that it depends. It's not always an image and then treat approach that needs to be taken. So, for example, in our first program in small cell lung cancer, we know that from, the target that we're proceeding, targeting DLL3, that's expressed at a very high consistency in small cell lung cancer tumors. And so with 80 to 90+% of patients having their tumors express that target, it's not necessary to have to always image and then treat those patients. But it's certainly helpful to be able to validate that there is uptake in the tumor. But in patients, particularly in indications where the patients are quite advanced when they're going to be getting this therapy, it's a fast progressing disease. We also thought about balancing what would be the best thing for the patient, and which is that in our first study, we won't necessarily require that all patients are imaged and then treated because of the expression level of the target. If you were looking at a target that's perhaps expressed on 40 to 50% of the cancers, you may want to proceed to do an image then treat. So what I would say is it probably depends upon the target that you're engaging with your, therapeutic agent. But we'd love to hear what others think about that.
Yeah, I think in the case of PSMA, we use PSMA PET scans in advance, but most patients, probably 90% express PSMA and don't lose their expression even after they're treated with Pluvicto. So one of the advantages of using an antibody and using alphas is that it's effective even in the very low expressing PSMA patients, more so than, so patients who have failed Pluvicto will respond to what we call Convo 1 alpha, which is the antibody alpha approach. With regard to dosimetry, we're learning right now. We're doing indium scanning in patients before we treat, just to reassure ourselves that it's going to the right places. And George Seguros, who's one of the experts in the field of dosimetry, particularly with alphas, is working out dosimetry for alphas right now, which is very exciting. So you can actually image alphas, which was previously, at least actinium, previously not doable. So dosimetry is a very exciting part of what we're doing right now.
I would say the imaging aspect doesn't just necessarily de-risk the program, but it also allows you, once you've developed an indication, to look in other indications for expression. For example, we know that somatostatin 2 receptor, which is the Lutathera GEP-NETs kind of product, is actually expressed. That target is actually expressed in a range of other cancers at lower levels. So, for example, small cell lung cancer, some breast cancers, neuroendocrine type prostate cancers. So one can envisage sort of expansion into additional indications on the basis of additional imaging for patients that do express the tumours. I can envisage a world where down the track, clinicians will have a suite of different traces and the associated therapies that they would use to screen patients on the basis of target expression, and then select the right RLT to treat the patients.
I think that one of the most interesting times that's coming up in the next couple of months is whether CMS is going to change the way that they allow PET and SPECT imaging agents to be reimbursed. I think there's the reason, in part, why I think PSMA and SSTR2 became targets was there was a lot of homebrew use of them. There were companion diagnostics that were developed so you could look for the right target avid patient population to treat. But in the U.S., the way that reimbursement worked is you got reimbursed for your PET imaging agent or your SPECT imaging agent for 3 years, and then it became bundled as part of the imaging, the procedural cost itself. There was a CMS ruling, I think Lantheus was one of the beneficiaries of this, in which they allowed them to continue to be reimbursed for their imaging agents. Again, back when you were using FDG, this made sense, but these imaging agents are far more expensive. I think the interim rules have come down, and they are expecting final rulemaking in November 4th, I think is the deadline, the date for it. And if at that point CMS determines that they will reimburse for imaging agents that are above $650, it was a $600 threshold, I remember, I think that will open the door for companies, whether it's companies like us on the stage or companies like Lantheus and others out there in the diagnostic space, to really be incentivized to develop the imaging agents that would be used in patient stratification and following patients after treatment.
Go ahead.
So the toxicity is minimal compared to a number of other therapeutic approaches, and it's orthogonal, so the toxicity profiles tend to be somewhat different. So how has that informed your thinking in terms of combinations?
Well, we know that, for example, alpha particle therapy is extremely immunogenic, and so what happens is when the alpha particle rips through a cell, it generates neoantigens that are relevant to the patient and their tumour. So what that means is that if you can wake up the immune system in the presence of those neoantigens, what you're actually doing is generating a vaccine-like response in the immune cells. So at Perspective, we really believe in this approach. We're doing a trial on it, essentially, and I think that's probably what gets us most excited about this space, is that we've really got the opportunity to drive durable responses. It's not just cytoreductive, it's also immunostimulatory, so the combination is a key part of what we're doing moving forward. I'm not sure about
I just add that that's right now still a theoretical consideration. Certainly preclinically, the data would support it. We have treated some patients with a combination of Convo-1 alpha, the antibody actinium, with Pembro, and patients with far advanced prostate cancer, and we've seen 3 out of 6 patients at a higher dose of a single dose of Convo-1 alpha having durable responses, 3 out to 2 years without PSA progression, and 2 beyond 3 years. So there may be something to it. UCSF has also seen some durable responses when Pluvicto has been used in combination with a checkpoint inhibitor. So, there are some clues here, but I'm not completely convinced that the combination is real. And to say that there's synergy, I think it's a really interesting theoretical consideration and needs to be proven out.
Sure. I would add maybe that if you look at the data with targeted radiopharmaceuticals, it's very clear based on the mechanism. We understand very well the underlying mechanism of action. And so we absolutely expect, I believe everybody would, I don't think this is going to be controversial. I think we all expect that there's going to be robust single agent activity with these molecules. It's kind of why we're here, right? But on the other hand, one of the things that's remarkable about targeted radiopharmaceuticals is how well tolerated they are. In fact, if you look at both Pluvicto and Lutathera, approved products in the space, check out the Kaplan-Meier curves, not only in the OS and the PFS, but also the quality of life, right? And one of the really nice things about this, this modality is the tolerability, the quality of life gains that you get from this approach. Now, that does leave open the door of the combination therapy, as you're suggesting, and I think all of us are compelled. We are responsible for pushing forward those experimental approaches to determining what the best combinations are. But I would also, I don't want to ignore the fact that one of the reasons that there is quality of life associated with this is that not only do you get the efficacy, you get depth of response, you get durability of response, but you also get a treatment break, which a lot of cancer patients and their families really appreciate.
Yeah. I think the other thing that's going to be required for combination is dose optimization first, right? I think you're going to be looking, that's going to be this is our second generation RLTs up here. The second or third generation of dosing regimens, it really has to start focusing on the doses you're giving, the duration between doses. Again, if we think that there's an immune component to this, it's priming and constantly re-engaging the immune system, maybe more frequently than we currently do. A lot of that's going to come down to isotope supply as well, but I think dose optimization will eventually lead into an understanding of what are the right tumor types and the right drug RLT opportunities that could support a combination based on what you see in different dosing cohorts that will start coming through the clinic.
Yeah, I think that's completely right. I agree with that, because when you look at it, it's only really been about 10 years since, I would say, the first next generation radiopharmaceuticals like Vigo was approved, and then we had Lutathera in 2018, and then you have Pluvicto. And so we're early in the curve of this next wave of innovation in radiopharmaceuticals. And if you look at the drugs that are getting approved or going into the clinic, we're all, it's all around the same really dosages of activity that we're delivering, and that's the right place to start. And obviously, as the regulators get more comfortable, as we collectively build the data sets and understand how we can improve the dosing, there's just a lot of activity that needs to happen. So that's why we learn across, even though our approaches and our delivery systems, whether they're large molecules or small molecules or somewhere in between, at the end of the day, we're all trying to deliver alpha and beta particles right now, which is the active pharmaceutical ingredient. So whether or not, Matt decides he's going to use a mini protein to deliver it, if it's actinium-225, we're learning about the dosing and some of the dosing regimens. And so that's where there's such a collective energy. We don't, there's the delivery system, which is what we're all developing, but it's the radioisotopes that are the active agent. And so we all learn a lot from each other. So much more to do on the dosing regimens, for sure.
Look, unfortunately, we've only just scratched the surface on this, and there's a lot more to discuss, but we're encouraged with the progress thus far, and we look forward to seeing the progress for each of your companies, and wish you the best of success. Thank you for joining us.
Thank you all for listening.
Thank you.
Thanks, everybody.